The Stockholm Concert Hall gleamed in the December sunlight, its neoclassical façade a testament to timeless excellence. As I prepared to deliver my Nobel lecture, I couldn't help but reflect on the journey that had brought me here, a path marked by betrayal, persistence, and the remarkable tendency of science to reveal its secrets to those patient enough to listen. 5 years earlier, I had stood in a very different setting, the emptied laboratory of Mitchell-Rhodes Pharmaceuticals, watching as security guards carried out boxes of research materials. Daniel Mitchell, my research partner of 8 years, supervised the process with clinical efficiency. "The partnership agreement is quite clear, Dr. Rhodes," he had said, his voice carrying that familiar note of academic condescension. "All mainstream research materials and patents belong to the majority shareholder. You can keep your side projects and failed experiments." Those failed experiments consisted of years of research into what I believed was a revolutionary approach to treating autoimmune disorders. The preliminary results hadn't matched our expectations, leading Daniel to dismiss the entire line of inquiry as wasteful. But I had seen something in the data, subtle patterns that suggested we were looking at the problem from the wrong angle. "You're welcome to them," he had added with a dismissive wave, "though I doubt they'll help you recover from bankruptcy." He wasn't wrong about the bankruptcy part. Our company's financial troubles had given him the opportunity to invoke clauses in our partnership agreement that effectively stripped me of everything except my personal research, the projects he had deemed worthless, the mainstream research, the promising drug candidates, the valuable patents. All of it now belonged to him. I had watched them dismantle the lab, carefully noting which materials they took and which they left behind. Daniel's team was thorough in claiming anything related to our conventional drug development program, but they ignored the cabinets containing my autoimmune research materials. Their oversight would prove monumentally significant. That evening, after everyone had left, I sat alone in the nearly empty lab. The remaining equipment and materials represented less than 5% of our original resources, but they included something priceless, the complete dataset from my supposedly failed experiments, along with all the biological samples and preliminary compounds. I opened my laptop, one of the few pieces of equipment that was legally mine, and began reviewing the data for the 1000th time. The patterns were still there, teasing at the edges of understanding. The drug compounds we had developed weren't failing. They were doing something entirely unexpected. Traditional autoimmune treatments focused on suppressing the immune system's overactive response. My compounds appeared to be doing something different. They were modulating the immune system's behavior in ways our standard assays hadn't been designed to detect. I worked through the night, cross-referencing data points and reviewing cellular response patterns. By morning, I had formulated a new hypothesis. What if we had accidentally discovered a way to reprogram the immune system's recognition protocols rather than simply suppressing its activity? The implications were staggering, but proving the hypothesis would require resources I no longer had: the sophisticated testing equipment, the funding for clinical trials, the support staff. All of that had been stripped away along with my professional reputation. I began reaching out to former colleagues, but Daniel had been thorough in poisoning those relationships. Dr. Rhodes is pursuing outdated theories, they had been told. Her inability to accept negative results led to our company's financial difficulties. Most doors closed before I could even present my new findings. The few who did listen were sympathetic but unwilling to risk their careers on what seemed like a desperate gambit by a discredited researcher. Finally, I contacted Dr. Sarah Chen at Stanford's Institute for Immunity Research. She had been a mentor during my postdoctoral work, and her integrity was beyond question. More importantly, she had always emphasized the importance of following unexpected research outcomes. "Send me everything you have," she said after listening to my explanation. "And I mean everything, especially the failed trials and anomalous results." I spent 3 days organizing my data, ensuring every experimental detail was properly documented. The package I sent her included not just the successful trials, but every failure, every unexpected result, every observation that didn't fit our original hypotheses. 2 weeks later, she called. "I need you to come to Stanford immediately," she said, her voice tense with suppressed excitement. "Bring any remaining samples you have. And Catherine, don't talk to anyone about this." The flight to Stanford felt like crossing a threshold. Behind me lay the ruins of my career and reputation. Ahead, perhaps, lay vindication, but more importantly, ahead lay the possibility of turning a scientific failure into something that could help millions of patients. Sarah met me at her lab, her usual professional composure barely containing her enthusiasm. "Look at this," she said, leading me to a microscope. We replicated your experiments using more sophisticated imaging technology. Watch what happens to the T cells when exposed to your compound. The images on the screen showed something remarkable. The immune cells weren't being suppressed, they were being reprogrammed. Their surface proteins reconfiguring in ways that suggested a fundamental shift in their recognition patterns. This isn't just another immunosuppressant, Sarah said, her voice filled with quiet awe. This is something entirely new. You found a way to teach the immune system new rules of engagement. We worked through the night, reviewing data and designing new experiments. By morning, we had outlined a research program that would properly investigate the mechanism I had accidentally discovered. We'll need funding, Sarah acknowledged, but I have connections with several research foundations. Once they see this preliminary data. She paused, studying me carefully. Catherine, do you understand what you've discovered here? This could revolutionize the treatment of not just autoimmune disorders, but potentially allergies, transplant rejection, even certain types of cancer. I nodded, my throat tight with emotion. I know, I've known for years, I just couldn't prove it with the tools I had. Well, she smiled, now you have the tools, and more importantly, you have a partner who can see the value in failed experiments. The next few months were a blur of grant applications, experimental design meetings, and careful documentation of our research protocol. Sarah's reputation opened doors that had been firmly closed to me, and her institute's resources allowed us to explore aspects of the mechanism that I had only theorized about. Each new experiment confirmed my original hypothesis while revealing additional layers of complexity. The compounds weren't just modulating immune response, they were establishing new immunological memory patterns that could potentially create long-term disease remission. As our understanding grew, so did interest from the medical research community. Preliminary papers were published, conferences invited presentations, and funding began to flow more freely. Daniel, meanwhile, had taken Mitchell Pharmaceuticals in a more conventional direction, focusing on incremental improvements to existing drugs. The company was profitable but unremarkable, producing nothing that could be considered genuinely innovative. He remained unaware of our progress, having dismissed my research so completely that he didn't even monitor publications in that field. His focus was entirely on the mainstream drug development programs he had acquired in our split. The breakthrough came 18 months after my arrival at Stanford. Late one night, as Sarah and I reviewed the latest trial results, we saw something that made us both catch our breath. The long-term follow-up data showed complete disease remission in 94% of test subjects, with no sign of relapse after 2 years. This is it, Sarah whispered. This is what medicine has been waiting for. I nodded, thinking of all the patients who might benefit from what had once been dismissed as a failed experiment. Sometimes, I realized, the most important discoveries come not from success, but from paying attention to what others consider failure. The publication of our findings in Nature Medicine created immediate waves throughout the medical research community. Our paper, titled Novel Immune System Reprogramming in Autoimmune Disease Treatment, presented comprehensive data demonstrating the mechanism's effectiveness across multiple conditions. Within days of publication, our laboratory received inquiries from major research institutions worldwide. The data was revolutionary, showing not just symptom suppression but actual disease remission through targeted immune system reprogramming. The WHO committee wants to fast-track clinical trials, Sarah announced one morning, reviewing her emails. They're calling this a potential paradigm shift in autoimmune disease treatment. The significance of our discovery became increasingly apparent as other laboratories replicated our results. The compound wasn't just effective, it was remarkably safe, with minimal side effects compared to traditional immunosuppressive therapies. Daniel Mitchell finally became aware of our work when the pharmaceutical industry's major players began approaching Stanford about licensing opportunities. His attempt to claim ownership of the research through our old partnership agreement was swiftly dismissed. He had explicitly relinquished rights to these failed experiments in documented correspondence. The Phase 3 clinical trials exceeded even our optimistic expectations. Patients with severe rheumatoid arthritis showed complete remission. Multiple sclerosis progression halted and reversed in 89% of cases. Lupus symptoms disappeared entirely in 92% of participants. The FDA granted accelerated approval based on the overwhelming evidence of efficacy. The first commercial treatment, named Immunoplex, became available to patients 2 years after our initial publication. Letters began arriving from patients and their families. A young artist with rheumatoid arthritis could hold her brushes again. A teacher with multiple sclerosis returned to the classroom. A teenager with severe lupus was planning for college instead of managing constant hospitalizations. Dr. Rhodes. One letter began. For 20 years I lived with pain that no one could cure. Your medicine gave me my life back. Thank you for not giving up. The pharmaceutical industry took notice. Major companies competed for licensing rights, leading to a bidding war that ultimately resulted in a partnership that ensured broad access to the treatment while maintaining research integrity. Sarah and I established the Rhodes Institute for Immunological Research, funded by licensing revenues and dedicated to exploring additional applications of our discovery. We assembled a team of researchers committed to investigating unexpected results and challenging conventional wisdom. 3 years after our initial publication, the call came from Stockholm. The Nobel Committee had selected our work for the prize in physiology or medicine. The citation praised not just the scientific breakthrough but the persistence in pursuing promising results despite institutional skepticism. As I prepared for the Nobel ceremony, an invitation arrived from an unexpected source. Daniel Mitchell requested permission to attend, expressing what appeared to be genuine remorse for his earlier dismissal of my research. You should invite him, Sarah suggested. Success is the best revenge, but forgiveness might be even better. The ceremony itself was surreal. The grand hall was filled with the world's leading scientists, dignitaries, and medical professionals. In the audience, I could see Daniel, his expression a complex mixture of regret and admiration. My Nobel lecture focused not just on the scientific breakthrough but on the importance of questioning apparent failure. In science, I explained, what we initially perceive as failure often contains the seeds of unexpected discovery. Our responsibility is to look deeper, to question our assumptions, and to remain open to possibilities that don't fit our preconceptions. The data I presented showed the expanding applications of our discovery. Beyond autoimmune diseases, the technology was showing promise in treating allergies, preventing transplant rejection, and even modulating immune response in cancer treatment. After the ceremony, Daniel approached me. I was wrong, he said simply. I was so focused on conventional success that I couldn't see the revolutionary potential in front of me. The science was always there, I replied. It just needed someone willing to look past apparent failure to see the truth. Our conversation was brief but significant. He had achieved moderate success with conventional drug development, but nothing approaching the impact of what he had dismissed as worthless. The Rhodes Institute continues to expand our understanding of immune system modulation. We've established a special division dedicated to investigating unexpected experimental results, ensuring that no promising discovery is overlooked due to preconceptions about success and failure. Today, as I mentor young researchers, I emphasize the importance of questioning apparent failures. Look deeper, I tell them. Sometimes what seems like a dead end is actually a new beginning. Your job isn't just to conduct experiments, it's to listen to what the data is really telling you, even when it doesn't match your expectations. The original laboratory notebooks from those failed experiments now reside in a special collection at Stanford's Medical Library. They serve as a reminder that scientific progress often comes not from following established paths but from having the courage to explore unexpected directions. Last month, I received a letter from a young researcher at MIT. She had encountered resistance to her unconventional approach to protein folding analysis. Your story inspired me to persist, she wrote. Thank you for showing that today's failed experiment might be tomorrow's breakthrough. The letter now sits on my desk at the Institute, next to the Nobel medal and the first patient thank you note we received. Together, they remind me that science is not just about being right, it's about being willing to look deeper when others have stopped looking. Daniel occasionally sends updates about his work, and we maintain a cordial professional relationship. He's become an advocate for investigating unexpected research outcomes, having learned firsthand the cost of dismissing potential breakthroughs. The Institute now hosts an annual conference on revolutionary failures, bringing together researchers to present unexpected results that might contain hidden breakthrough potential. It's become one of the most innovative gatherings in medical research, generating collaborations that cross traditional boundaries. As I conclude each year's conference, I share the same message. In science, there are no real failures, only results we haven't properly understood yet. Our job is to keep looking, keep questioning, and keep believing in the possibility of discovery. The Nobel Prize was validation, but the real reward comes from patients whose lives have been transformed by our discovery. Their stories remind me that scientific breakthrough isn't just about professional success, it's about making a difference in people's lives. That empty laboratory where Daniel left me with my failed experiments is now recreated in a display at the Institute. It stands as a testament to the power of persistence and the importance of looking beyond conventional measures of success. The future of medical research lies not just in well-funded laboratories and sophisticated equipment, but in the willingness to question failure and explore unexpected possibilities. Every day, somewhere in the world, a researcher looks at disappointing results and faces a choice: accept failure or look deeper. I hope they look deeper, because sometimes, in what others dismiss as worthless, lies the key to changing medicine forever.